Hi, my name is Ian Donaldson. I'm going to take you through a brief overview of iRefIndex and its related resources. In a nutshell, iRefIndex is a consolidation of about 10 different interaction databases focusing on protein-protein interactions. It takes these databases and consolidates them into an iRefIndex build database. In addition, it provides feedback to the source databases on problems that we experience during the build process. The iRefIndex build database is then used to make three other consumables that can be used by users. First of all is the PSI MI tab 2.6, which is available from our FTP site. Then there's the iRefScape plugin for Cytoscape. And finally, iRefWeb database, which drives the iRefWeb application at the Wodak lab. These two resources here are available from iRefIndex.uio.no. The Cytoscape plugin can be downloaded from Cytoscape itself, and the iRefWeb resource is available from the wodaklab.org iRefWeb. So the types of information that we are talking about here basically capture information about interactions between proteins, where we record short labels, the type of molecule, the database identifier, and the origin for each of the molecules involved. Again, we're only interested in proteins. And information, meta-information about the interaction itself, and that includes the detection method, the interaction type, the publication reference, and the database identifier, as well as a lot of other information. The types of information are shown with examples here. TRAD is a short label. In all cases, we'll be interested in proteins. Uniprot KB is a type of accession used to refer to that protein, and it comes from human. This particular interaction may have been shown by yeast 2 hybrid and supports the idea for a physical interaction. It comes from this publication reference and is found in this interaction database reference here from Intact. For historical reasons, there are a large number of interaction databases that all started up in the early 2000s. The ones that we integrate are shown here, with the exceptions of MPIDB and InnateDB, and MatrixDB, which will appear in release 8.0 of iRefIndex. All of these databases are part of the IMEX consortium, which was formed around 2005, and this consortium does a number of things. First of all, they exchange data with one another and share curation tasks. They split up the curation of various journals. They set standards, and they also accept interaction data submissions pre and post publication. Data consolidation and exchange is an active area of development right now, most obvious in the Cycwic services development. You can find more information about IMEX at the imexconsortium.org. This is the paper showing the level 2.5 of the XML standard HUPO-PSI format for molecular interactions, where they describe the data exchange standard. In addition, this group also takes care of the molecular interaction ontology, which you can browse through at the ontology lookup service. We also incorporate a number of other interaction databases outside of the IMEX consortium, including Quorum, HPRD, MPPI, and OFEED or I2D. There are many others that we don't incorporate at the moment, and those can be found at pathguide.org. To go back to summarize the integration process, we take in about almost 1 million protein-protein interaction records, and in a process of assigning those to redundant groups, which I'll cover in a separate video, we're able to make those assignments in almost 99% of the cases. However, the number of distinct interactions that are actually available in all of these records is only 433,000. So there's a certain amount of redundancy where we describe a distinct protein interaction as being some sort of relationship shown by any kind of experimental method between the same two proteins. All of the resources that I'm going to talk about in this video and subsequent videos are available from iRefIndex.uio.no. You can find a richer set of statistics at this page here, by clicking on this link. There's also access to the web interface, the iRef web interface, the Cytoscape plugin, and web services available from this site. And the data availability via download from the FTP site is also linked from this site here. Just to quickly go over some of these other resources and introduce them, this is what the iRefScape plugin for Cytoscape looks like, and allows you to have access to the data that's in iRefIndex. In addition, there's a web interface that's been developed in collaboration with the WODAC lab, and this interface provides a very rich set of data filters to help people look for the interactions that they're interested in, and it's probably the best way to find a subset of the MyTab files interactions that you're looking for. You can do a search here and then download the interactome or your set of search results using this button here. Well, finally, iRef Web also provides the ability to cross-compare curation of the same paper by several different databases. So across these columns here are several different databases, and down these rows you can see several different interactions, and in this instance here there's one binary interaction that has been observed by both Bind, BioGrid, and DIP from the same paper. Finally, iRefWeb also provides a set of rich statistics on their site. It's good to keep in mind that these numbers may differ somewhat from the iRefIndex statistics because these exclude predicted interactions and genetic interactions when calculating statistics. Finally, iRefWeb also provides a web-based, Cytoscape web-based interaction viewer to the data. So in subsequent videos I'm going to go into more detail about the data consolidation process used by iRefIndex, and then also specifically talk about representation of complexes and go into a little more detail about iRefScape and iRefWeb themselves. These are the people that are responsible for iRefIndex and iRefWeb. In my group, Sabri Razik primarily, and Shoshana Wodak's group at the Hospital for Sick Children.